Okay, so today I wanted to talk to you guys about using naturalistic approaches in fMRI. And so I'm going to focus mostly on using natural vision. That's where most of these studies have been taking place, but there's an increasing amount of studies using audition as well, particularly using like stories from NPR or Storyfy and stuff like that, where they're presenting these auditory stimuli and these auditory stories, or even having subjects talk to each other in the scanner type of thing. So this isn't just about vision, this is more about using more naturalistic approaches and the trade-offs that come along with that. So to go into that, I'm going to start out talking a little bit about visual experience, how that interacts with paradigms that you're using, what those design choices mean for the experiments that you're going to end up running. And then the really meat and potatoes of it is how do you actually get information, experimental results out of these studies. Because the secret is it's very easy to run a naturalistic study. It's very easy to just have a subject lay down in the scanner, show them a movie, and collect that data. That's not the hard part of these experiments, whereas in many cases the hard part is programming up a really complex design, but then the analysis is really easy because it's basically run a t-test between your two conditions. Here, the complexity comes from trying to pull out signal from a very, very complex response that you're going to be getting, especially when you're dealing with fMRI that has these long time scales. So we're going to spend some time talking about how different ways that that's been done, and then I'll just try to summarize some stuff. Feel free to ask any questions along the way, clarifying or otherwise. I'm happy to answer them. Otherwise, we'll get into it. So starting off with visual experience, the brain has to take in a very complex world. So shown here is a painting. Everyone can look at this painting real fast and see that this is a waterway. There's some boats, people walking along a bridge here. You can see people in the water back here. The thing is, you probably all perceive those as people, but if you zoom in on this photo, what you'd see is literally it's just a line of paint with a dot above it, right? But we very quickly interpret that as a person because our brain is built to take in all of this information very quickly and make judgments about it. And so we all very quickly interpreted the meaning that the painter had in this case, and figure out what they meant as opposed to what exactly is on the screen. We don't view our world in a very veridical manner, meaning we don't just see the dots, we see the whole gestalt image that's intended. And we do this through a very, very complex system. So information hits your retina, goes through the optic nerve, is processed in some low-level areas, gets sent up to your visual cortex. That's in the back of the brain. Then that's getting sent that information is going to go all along the visual pathways, covering your temporal lobes, into the dorsal stream, eventually ending up in your frontal cortex, where you're probably going to make some kind of decision about what you saw, and then that information is going to get sent to your motor system. So nearly the entire brain is involved in vision, essentially. It's involved in your other senses too, but estimates range from somewhere on the order of like 40 to 70 or 80% of your brain can be involved in vision depending on how you want to define that. So clearly a lot of processing power is going into this. Now the thing is, it needs all of it because the brain isn't just seeing this static picture every time. What it's really seeing is scenes like this. So I tend to work with macaques, so I'm going to show you a macaque movie here. But we see these animals interacting with each other. There's some aggression going on, a little bit of grooming. They're essentially talking to each other through facial movements. And all of this is happening in real time, right? And this is what's happening all the time for us. Things are moving through our environment. When we're driving a car, we're moving at 55 miles per hour. The people around us are moving at slower and faster speeds. All of that's impinging on our retina at all times. And our brain has to not only take the individual static frames, but keep track of what was going on in the past. So this is kind of how the real world works. It's dynamic, it's not static, and this adds even more complexity to the information than we get even with just a static frame that we saw before, which was already extremely complex. In addition, we're going to add some more complexity on that. not only is the world moving around us, but we're moving through the world and we're collecting information in different ways. So a good example of that is all of you out there right now are listening to this talk and watching it. Some of you at any given moment are looking at me, some of you are looking at the screen, some of you are looking at the other participants in this thing, and each time you're each gathering different information than the person next to you. So a good example of that is I'm going to show you a short video here. The lines on the screen are a single monkey's individual eye movements during repeated viewings of the same movie. So this same clip was shown to the monkey 12 to 15 different times. Each color on the screen represents a different trial that the monkey was watching this. So if you notice, their eyes are kind of all over the place here. Each movement here is a different trial. I think we're only showing 2 trials here, but I've done this for 15 different ones. And you can see that at all different moments are kind of moving all over the place, right? And so each time they viewed this, they got a slightly different visual scene that occurred as opposed to the last time. And so the brain has to account for that as well. So even though you may have watched Braveheart 20 different times, random movie pulled out there, but most of you probably have seen it way too many times. You see this movie and you remember the whole thing, but each time you watched it, you probably looked at a slightly different feature than you did the time before. And you still got the full gestalt of it because your brain is good at pulling in this extra information, and it's good at holding information over time. But this adds a lot of complexity to how your brain is processing this stuff. So this gets us to trying to understand our experimental design choices that we make. So basically, most experimental designs are a trade-off in how we deal with this complexity. Some people may not think of it that way, but essentially that's the way it's going to end up working. So one way we can think about it is a continuum of artificial but extremely well-controlled studies all the way to the natural world around us, but essentially losing all control that we might have over the stimulus. And there really are advantages and disadvantages to both. The question becomes, what is your question and how is the best way to answer it? So for some people, the best way is to get as much control over the world as possible because they have a very specific question they're trying to ask. So a lot of early neuroscience, visual neuroscience was done this way. You might anesthetize an animal, you prop open their eye, and you show visual gratings to the animal while they are essentially asleep. And you get a lot of control over the stimulus. The eye is not going to be moving. You can specifically target a very specific portion of the retina because you're sure that everything's going to go where you want it. You can show the stimulus for as long as you want, and you get tons of control. But what you're lacking is ethological validity, right? This is not a stimulus that's showing up a lot in our everyday world as we're moving through it. And additionally, when they're anesthetized, that's a very different brain state than an awake animal moving through the world. So we can try to relax it. We can go all the way to the other side. We can go to, say, in some advanced number of years, maybe we get to the point where we can implant a array of electrodes in a bunch of animals, have them living in the wild. We're recording telemetric data from them, so we're getting constant data from a whole bunch of areas, from a whole bunch of animals as they're doing all sorts of different tasks. The problem with this is it's extremely uncontrolled, and in this case, in today's day of being able to analyze this, in the end, we'd actually be able to tell whether the animals were asleep or awake because the data would just be too complex and we don't have the current ability to necessarily interpret it using the correct algorithms or thoughts, but maybe we'll get there someday where that's a useful paradigm. So most people end up sitting somewhere in the middle of these two extremes. I mean, a lot of stuff goes on the far left extreme still, but everyone's kind of moving towards a more middle ground. The position of this dot is kind of made up, but let's just say we move a little bit away from completely artificial. We're now dealing with an awake animal, say a human in the scanner. One thing that people tend to do to get a little bit more ethological validity, they start using more naturalistic stimuli. For example, instead of using a grating or a circle, they're going to start using things like faces, right? And we're going to understand how the brain might process faces. And so we do that in a standard block design. We can show faces versus scrambled faces or faces versus objects. Lots of studies have been done this way. There's lots of different little nuances in how you can do it. You can do it in event-related designs. You can do it in a block design. You can vary the amount of information that the subjects get to see. But in the end, you're kind of getting really tight control. You're going to show a face for X amount of time. You're going to show another stimulus for X amount of time. And then in the end, you're going to compare those stimuli to each other, the responses to each other. You get a time course for every voxel in the brain. Looks something like this, a little bit noisy, but it's going up and down based on the stimulus you're presenting. And then you can basically turn those time courses into a map of activity when it's high for one thing, low for another. And so here's some data looking in the macaque at faces versus objects. And what you see in red and yellow are what are called the face patch system. And those are areas that respond more to faces than to other stimuli. Surrounding them in the blue are object areas. Below those, this paper would tell you that there's also color patches. And so there's a lot of different ways that we can do this. In humans, these same types of studies have been used to find areas like the fusiform face area, the occipital face cortex, those types of things. And so we gain a lot of information on this. We start to learn what areas of the cortex are involved in processing particular categories of stimuli. And then we can dig in a little deeper as we start learning that and ask more nuanced questions about individual areas. Do these ones care more about identity? Do these ones care more about shape? That kind of thing. So there's a lot of ways you can dig down into this, but you're still going with a very controlled stimulus that's not very ethologically valid because you're only really asking one question. Does this voxel like this better than that, right? And that's how most of these block designs work. And so the question is, can we get a little bit more further along the lines and get a little bit more ethological validity while not losing complete control of the stimulus? So this dot might be a little too far, but one way to do that is to take these videos that I've been talking about and start showing them to the animals or humans in the scanner, and you lose some control. So now the animal's allowed to look wherever they want instead of fixating on a dot. There's a lot of motion in the scenes. They may be looking at one thing one time and another thing another time. And this adds, you've lost some control over your behavior and your stimulus, but you've gained a lot in the sense that this is much closer to what the brain has evolved to process than, say, a grating or just a static face on the screen flashing into a new one. Because I don't know how often this has ever happened to you, but I've never been walking around and had a face randomly change into a different face, which is a lot of what happens in these block designs. But I have been in a lot of instances where, well, maybe you guys haven't, but I've been in a lot of instances where these monkeys are fighting in front of me. But so one way that this has been done, kind of classic example of it, is the good and the bad and the ugly. So a lot of these studies using more naturalistic stimuli started with Uri Hassan and Rafi Malik back in around 2001-ish. They did a study where they compared different subjects' viewings of the good, bad, and the ugly. They watched a whole chunk of the movie, and then they compared their brain responses either between subjects or also just looking for what features in the movie caused responses in particular areas. So that group kind of started off this chain of more recent studies that have grown exponentially over the years, looking at more naturalistic videos. Just going to go back a second, re-show that. And so, there's still unnatural bits to this, right? There's zoom in on eyes, there's these hand shots, there's a lot of cuts. It's not the natural world. It's not just a static world that we're moving through, but it's much closer. This is much more realistic than just flashing images at a subject. And so we gain a lot of validity, but now we have to figure out how are we going to understand what actually happened during that. And so moving forward, I'm going to start talking a little bit more about the actual analyses that go into trying to understand these responses. So one way of doing that, and a lot of different groups have come up with this, I'm going to start by focusing a little bit on some of the stuff we've done here and then expand to some of the other studies that have done it, is to try to code the different features within the movies and then use those in some form of a regression to try to understand how the brain is processing these given features. This leads to a lot of complexity. Specifically, you have to deal with a lot of intercorrelation. So one of the good things about doing something like an event-related design or block design is that those designs, if you've written them properly, there's no correlation between your images, right? So you've specifically set up your images in a way that your directions of study are orthogonal to each other. So if you have like two different conditions that you care about, you're going to make sure that they're not correlated with each other in the experimental design. In the real world, that's not very possible. So let me bring up a few of the features that we've gone through. So in this case, we've coded for a lot of different features. We coded for luminance, motion, the spatial frequencies in the scenes, the contrast. All of these features, they're easy to get because there's algorithms you can run on your stimuli to get these. Life gets a little harder when you start talking about more high-level visual features. So we can do animals, such as the number of, say, macaques in the scene or the number of humans in a scene. But then these things also, this is where these correlations come in. So humans and mammals are going to be correlated with each other because humans are mammals. So every time you see a human on the screen, you're going to have to code it as both a mammal and a human. So it becomes hard to tease apart whether an area might be coding for mammal versus human. And we're going to get into that in a little bit, how we try to go about doing that. Same thing for body parts. When you see butt, you're probably seeing a body. When you see a head, there's a face most of the time, but not all of the time. So there's these correlations built into the world that are really hard to get rid of when you're using more naturalistic stimuli. Now, in a controlled block design, you might be able to specifically design your stimuli so that you can actually differentiate head from face by very carefully parameterizing when you see a face with the head with the face forward versus the back of the head or the side of the head, that kind of thing. So there might be ways you can get tricky with that and decouple them from each other. But in the real world, it's a little hard to do that. We can even go as far as to code different behaviors of the animal. So when they're being aggressive with each other, with humans, when they're talking or driving a car, that kind of thing. And so we can code tons and tons of features. Then we can try to understand how that feature may map onto the brain. And so the way we do that most often is you create a time course out of the feature. So you get the ups and downs of when there was motion. So the amount of motion in a scene at every given moment, you calculate that. That gives you a time course. Or you can calculate when there was a face on the screen versus when there wasn't. That's going to give you a time course for the movies as well. And then what we can do is different forms of regression can provide you information about how that particular feature compares with the responses across the brain. So if we take example here, this is the response, this is the coding of face on the screen for 3 different movies. I used 5-minute movies, so this is 3 different movies. We've convolved the coding of the movie with the hemodynamic response, so now it's kind of been smoothed out to match what we would expect the brain to be doing. If there had been a face on the screen at a given time, we kind of smooth it in time to match what the hemodynamic response would be. So that allows us to then compare it to voxels within the brain. We can do this for every single voxel in the brain. In this case, I'm showing you the results from one voxel in one of the more anterior face patches in the monkey, an area that responds very well to faces when you do faces versus block design, faces versus objects in a block design. And what you see is the time course of this voxel in white very well matches the time course of the face variable that we had, suggesting that this voxel is probably responding to changes in the faces throughout the scene. So lots of different groups have done similar types of things. So here I'm going to go through one of the studies by Jack Gallant's group. The first author is Alex Huth. What they did was they showed a whole set of natural movies, and in that sense I just mean regular movies. They showed these to human subjects. They coded the movies for 1,700 nouns and verbs, things like walking, talking, text, cars and buildings. Whenever these things were happening, they'd put a 1 down if it occurred, 0, and then everything was filled with zeros where it wasn't happening. So you get these little hash marks for all the category labels that they had. And then just like I was talking about before, they've also got the fMRI responses to all the viewings of these movies from the subjects. So then what they did was they can take the bold responses and they can run a linearized regression on these and try to pull out what the weights are for each of these different variables for each voxel. And this provides a mapping, essentially, of how well each voxel is being activated by these various different 1,700 different codings that they did. And they can look at individual voxels. I'm just showing one particular example from their paper right now. This is a voxel from the I always get PPA is the paraplace area, parahippocampal place area. Sorry, always forget that one. And what they show here, this is an area that in a block design has typically been shown to respond more to objects or houses and scenes than to, say, naturalistic objects like a face or a body and stuff like that. So it's kind of the opposite of your face area. It's responding to the other stuff. And what they found was that they were able to see a similar weighting for for this voxel when they used their naturalistic movies. So the red areas here say that there was an increased response in this area when these particular scenes were on there, and I'm not sure if you can read them all, but like it's structures, artifacts, containers, devices, and then there's a decrease when things like a body part or food were on the scene, or mammals. So this very well matches with the previous data. So one could argue, well, okay, so we've learned nothing particularly new from this, right? But what's interesting is now you can combine this with all the other voxels and all the other variables that we have, and you start to see patterns that come out that you might not see when you view it from this like deep down level. So one of the things that they did was they had these 1,700 variables that they did. So they ran a principal components analysis on the variables across the voxels, and they found 4 particular principal components. These are basically a way of reorienting your data to try to pull out the most variance. Each principal component accounts for a certain amount of variance, and they are, by definition, these things are now orthogonal to each other. So this goes back to trying to solve that problem of correlated variables. You're going to basically lump correlated variables together into different principal components. And then in the end, if you can, you might be able to try to name these principal components. Sometimes you can, sometimes you can't, depends on how complicated they get. But what you can do then is you can remap it onto the brain, the scores from these particular principal components for each voxel. And what they found, this is a very complicated map, but one of the main things they found was that instead of seeing distinct patches of cortex that respond to a particular variable, what they found was that there was a more smooth transitioning of the coding of these semantic labels. So what they saw was that as you move, so like here's a good example. So in this 3-dimensional coding space of color, you move from the dark blue to the purples, to the greens, to the reds, back to the greens, to the purples, to the blues. And it keeps repeating this smooth transition across the brain, suggesting that there's these levels of analysis that keep repeating within the brain. And instead of being a distinct patch of cortex that's saying, on the face cortex, it's more that there's this smooth transitions throughout. But you might not see this when you simply ask two questions. Do I like faces or do I like objects, right? If you just go to one particular variable, which they can show you right here, you all of a sudden get distinct cortex, right? Because now we're only dealing with, say, animate versus inanimate objects in the first PC. Now you see distinct patches of cortex. But when you include these other variables into it, you see that it's much more smoothly transitioning because the world isn't just animate, inanimate. There's other pieces of information that map onto that same trajectory. So if you only have 2 dimensions or even 1, you get these distinct patches of cortex. But when you go into a more 3, 4, 5-dimensional world, which we live in, it gets more complicated. Following up on that, we ran a similar type of study. I showed you a little bit of the results before, but before what I showed you was that we could map the face voxels, right? The question is, though, how does all of that interact with each other? So before I just showed you faces, now I'm going to try to combine it with some other pieces of information. So what we saw when we ran a stepwise regression. So stepwise regression is another way of dealing with shared variance within your variables. What that does is it says that I'm only going to use a variable if it accounts for a certain amount of variance within the analysis. And I'm only going to use one variable at a time. So say in the first iteration of the study, in the first iteration of the analysis, faces accounts for 20% of the variance. I'm going to regress that, I'm going to regress that faces variable off of the initial data, and then I'm going to redo the analysis, but I'm going to do it on the residuals of your regression minus the faces. So now I'm going to compare, say I submitted 6 variables, now I'm going to submit the other 5 variables, but on the leftover variance. So any variance that was attributed to faces is now extracted from the analysis and cannot be attributed to another variable. So this automatically orthogonalizes your variables. The problem is, there are problems with this where you may account for variance with one variable that should actually be attributed to another, but in general it's a good job of at least separating the variables out from each other. And you can run it multiple times to make sure that it's not getting the wrong answer. But so what I'm showing you here is the total variance explained by our total model. We get upwards of like 50–60% in some areas. Areas that are not colored essentially are below threshold, meaning they're below a 5%. Actually, it's 1%. 1% of the variance is not explained in those areas. And that actually makes some sense. So, for example, our videos had no sound. And so areas in like the auditory cortex are not having any variance explained because we actually had no auditory stimuli. So what we can do is we can look at individual ROIs across the brain and see what particular variable is explaining the most variance in these areas. An easy one, MT, is a motion area, and unsurprisingly, motion is explaining most of the variance within that area. These are just good sanity checks to see these things working. But starting to get more interesting, what we found was that even in like early visual cortex and areas that are known to be more shape sensitive, the main variable that was actually attributing for most of the variance was motion, this dynamics within the scene. Even further interesting, if we go on to look at the face patch system, so areas that are really known to be object-oriented things when you use more block designs, what we see is that, again, motion is accounting for more variance than any other variable. Now, within the face patches, we do see the face variable now accounting for some of the variance, but motion is actually accounting for more than faces. And this becomes interesting when you start thinking about how face patches are defined. They're usually defined as faces activating that area more than anything else. But what we can see here is that motion is actually activating those areas more than the face variable. And when we look at the correlation between faces and motion, the correlation's actually essentially 0, suggesting that it's not just that the faces are moving that's causing this, because face motion and faces are not correlated with each other. So this suggests that there's something a little bit more going on in these areas, something that we want to probe a lot more in these studies. Another way of showing that is you can see the dominance of motion throughout the cortex. We can plot along the ventral bank and of the STS and the lateral surface of the IT cortex areas that are known to be visual areas in the macaque. And we see that motion is accounting for most of the variance throughout, with a slightly decreasing amount of variance explained as you go more anterior, but it's still higher throughout. These upward bumps here, these are the locations of face patches. So again, we're able to pick out these face patches using these types of stimuli, but when you use a more naturalistic design, you see other information is present there as well. Something that, deserves further probing. So as I was saying before, most of these studies so far have ignored where the animal or the human, I guess humans are animals, but where your subject is looking, right? And that involves a whole additional set of complexity because it changes what hits the retina at any given moment. So a study we recently published tried to get at that exact question, which was how does stimulus-generated motion differ from self-generated motion? Self-generated motion, in this particular case, being the eye movements that are occurring, causing sweeps of information across your retina. So to explain that a little bit further, we can talk about the two different reference frames that we're using. We have the screen-based reference frame. This is kind of what everyone thinks of when they think about a movie. You just have your individual scenes, and at each particular pixel in the scene, you can calculate a motion vector. This is essentially what I was talking about before when I was telling you that motion is accounting for most of the variance. This is that variable that I was using. But then there's also the retina-based reference frame. So in this case, instead of the movie staying static, we're going to pretend like the animal's eye stayed static. Now, this is not what actually happened in the study. The animal was allowed to move their eye around, but then in the analysis stage, what we've done is we've reoriented the scene so that now it seems like the eye stayed still and essentially the movie moved around. And we did this by calculating the velocity of each individual eye movement that the monkey made. So it essentially comes out to something like this. The eye is fixated on this upper right point, say this beach area over here. Then it saccades to the lower left, which essentially moves the frame to the upper right. And you do this over and over and over again, and you calculate all you calculate all your velocities for all of the scads, and you create a retina-based self-generated motion signal. And now we can compare the response between the self-generated motion signal and the stimulus-based motion signal. So just recapitulating what we saw before, we're doing this on a trial-by-trial basis as opposed to on an average response, which I'd shown you before. So here is the activity caused by video motion, the actual stimulus. And this matches very much what we saw on the average. Strong responses in motion-sensitive areas and the STS and IT cortex. Then we can calculate the activity caused by eye motion, and it actually activated a lot of somewhat similar cortex, but at a slightly lower level. But more importantly, it covered other areas that were not activated when we showed the video motion. In particular, we see this strong dichotomy between self-generated and stimulus motion, where early visual cortex, particularly foveal V1 and V2, are responding a lot to the eye movements, and then later areas like MTV4, the STS, are responding a lot more to the actual stimulus. And this makes a lot of sense if you want to think about how the brain has to incorporate these stimuli, right? So early visual cortex really works on a retinotopic frame, so it's going to care where the eye is moving towards, and it's going to care that a movement had occurred, whereas later visual cortex, it's much more oriented towards trying to understand the gestalt of the whole world. I care that an object was out there. I don't care whether it was in the upper left or lower right visual field. And that's what we're seeing here with these results. And so this suggests that one of the reasons we saw in our original study, we saw lower correlations with a lot of the variables in early visual cortex was because those are the areas that really care about when a eye movement's being made. But our later areas, our IT cortex, the STS, they have basically subtracted out the eye movement by that point, and now they're worried about the external world. So additionally, I've mentioned this a few different times about how dynamics within the scenes are really important. But another way to get at that other than motion is to try to understand how much coherent information is really important for any given area. So a good example of this is that one might expect that the retina doesn't care at all about what happened one moment before. Its job is to take photons that are hitting it and code those and send off a signal into the brain. And it's just going to do that every millisecond of the day. It's going to send a new signal saying yes or no that something's hitting me. And it's not going to worry too much about what happened beforehand. But in anterior temporal cortex or up in your prefrontal cortex, you're going to really care about what happened 10 seconds ago, because for me, as an example, let's use audition, which might be a little more clear. In order for you to follow a sentence, you need to have kept the previous portion of the sentence in memory as the new words are coming online. And so this can run to the point where you need to have 30 seconds, minutes, even hours of working memory, one could call it, in these cells or voxels in order to really comprehend what's going on in the world. And so one thing that these dynamic movies allow you to do is start to try to understand how coherent information becomes important across the cortex. So a really good example of that comes again from Uri Hassan's lab. And Christopher Honey, who's now at Johns Hopkins, is one of the really spearheaded a lot of these studies. But what they did was they took their movies, I believe this was The Good, Bad, and the Ugly again, and they shuffled the scenes at different time scales and then presented it to the subjects over and over and over again, a couple different times. And then what they looked for was how the correlation between viewings changed based on the amount of information that was coherent information that was being presented to the subjects. And so what they found was that essentially, as I was describing it, early visual cortex really didn't care whether you showed the full movie, longish shuffles of 30 seconds, medium shuffles of like 12 seconds, or even really short shuffles of 4 seconds. So 4 seconds is essentially around the average cut that would occur in a Hollywood movie. It varies from movie to movie, but like the average movie tends to have some form of a cut, either a reorienting of the view or a complete new scene every 4-ish seconds on average. So you might like zoom in on a face or zoom out to a wide shot, that kind of thing. And then, you get to your mid thing, that's like a shorter scene, and then long would be like almost a full scene that you might see within a movie. And what we see is that these early visual cortex really care about what's happening in the moment. So even at a 4-second shuffle, they're still responding really well. But then as you go out to more mid-level visual areas, like the dorsal stream here in the parietal cortex, or these like V2, V4 kind of areas, you start needing more temporal information to actually get the same response that you would have gotten. And so you need to see at least these 12-second long clips before you respond similarly. And then when you get to really very anterior areas within the brain, TPJ is an area that's known to be involved in attention, particularly social attention. Some even think it has to do with like social cognition and understanding of others. So it's a very social cognitive area. So those areas, and like FEF, an area in the frontal cortex that's involved in eye movements, they take much longer time scales. So they don't respond well to the 4-second shuffled scenes. They don't really build up their response well. But at the long shuffles or the full movie, they are responding the same. And so what this suggests is that as we move from posterior to anterior in the visual system, the system needs more and more visual information to respond. And that means it kind of has a working memory of some kind. It has these temporal receptive fields. You can do this in audition as well. It actually works really well there, because if you think about audition cannot happen without time. There is no snapshot of a word, right? Because sound is projected through frequency and time. And so if you take only one literal millisecond of time, you basically have nothing. You just have a couple frequencies, and that's not going to discriminate anything for you. You need the frequencies in time to tell you something about what words are coming. And so what they've done is they presented a set of spoken narratives, and they compared those to written words, and then they looked at the overlap between those. And what you can see is that when you get outside of A1 and very early auditory cortex, words actually activate very similar, written words activate very similar areas as spoken narratives. So you can get the temporal receptive fields. You can see that these language is kind of coded in the same areas. And so there's a lot of different ways you can start using these naturalistic videos to understand how temporal information is being coded within the world. A few other ways that these can be used. These are just some experiments that have been going on either in our lab or others. There's ways of looking at social content. So a bunch of different labs have done this, but a good example is some stuff that we've been working on, which is we showed a bunch of movies with animals in them. This is the response that we saw across the brain to viewing the same movie on repeated iterations. Then we can do the same thing with movies that have no animals in them whatsoever, but we try to control for some of the low-level things by showing movies that have lots of action in them. In this sense, it was often things like you see here. This was a scene of a hurricane that had been recorded where there were no humans or animals in the scene, but there's waves moving, there's trees blowing in the wind, and there's a lot of different versions of these types of things. And so overall, it looks like a very similar response to these two types of movies, but what you can do is you can compare the responses across the two types of movies, similar to how they did the written word versus spoken word, and what you can see is differences, a difference area where there's only really a strong difference between these in this middle of the temporal lobe type of area. And when you compare that to the location of known regions within the macaque cortex, what we see is that this social region here is really restricted to the middle face area of these macaques. So it's not the more anterior ones, which one might think would be more social. It's really these middle ones that are doing the difference between social and non-social. The anterior ones actually have very similar responses regardless of whether there were any faces in the movie or not. Again, suggesting that these areas might be doing something more complex when you ask a question that isn't just faces versus houses. Okay, so moving forward, I want to talk a little bit about some of the other ways that people have used these designs. Again, going back to Uri Hassan's work, there's intersubject brain. So there's a lot of different ways that people have used these movies to try to get at similarities across subjects. So one way to do that is to show the same movie or the same listen to the same story in the same subjects, and then compare responses across subjects to see what areas are responding similarly and differently across subjects. And this can explain some individual differences. So what you see here, complicated way of showing that you can do subject 1, you can do the same viewing of the same thing across subject 2, different thing, and then you get the comparison between all the subjects. And one of the things that they did this in different movies as well, and they found something that's really interesting, and I kind of have glossed over a little bit here, but want to bring it up now, is that the stimulus you choose still matters a lot. You can't just keep showing the same thing over and over and over again, because you're going to get locked into the same problem of using faces versus objects, which is that you're going to start asking questions about that stimulus as opposed to about the greater ethological world. So a lot of these studies have been done using the good, bad, and the ugly. Though that particular movie does do a good job of activating the brain of individual subjects. This shows a very high correlation between subjects for viewing this movie. But what's interesting was when they presented a number of different movies, 4 in this particular case, they found that the pattern of activation was actually different across the different movies, and that some movies did a really good job of activating the brain, for example, Good, Bad, and the Ugly, or Bang, You're Dead by Alfred Hitchcock, but others were much more sparse, suggesting that subjects were viewing these movies in very different ways from each other when we started looking at these other types of movies. And this is going to give you a very different answer to your question of what's happening if you start averaging across subjects then. So for example, the movie City Lights by Charlie Chaplin, a much older movie, it did not do quite as good a job of activating subjects' brains as did, say, Bang, You're Dead. So Bang, You're Dead did the best of all of the movies they showed. In particular, the ones that really matter for this are it activated a lot of social areas, suggesting that the subjects all kind of followed the movie in the same way. We're interpreting it in the same way, looking at the same faces and subjects and really grasping the meaning of the movie. Similar thing to Good and the Bad and the Ugly. It's not quite as broad a cortex, but all these social areas are kind of activated. TPJ, precuneus, some medial prefrontal cortex stuff. These areas are kind of coming online, suggesting some kind of social activation. But when you get to City of Lights, it's only the low-level visual areas that are actually being activated similarly across subjects, suggesting that at least in some subjects, they're responding to this movie in a very different way. They might have a very different interpretation of what's going on. This gets even more the case when they showed a video that was recorded in Washington Square Park, so there's no fancy cuts, there's no director kind of moving your attention to particular things by zooming in on a face versus a particular actor. This is just a segment of time from one day in Washington Square Park. And so what you see is that actually here, there's very little cortex that's similarly activated across subjects. So this goes back to, moving towards that more ethological stimulus. Yes, Yes, these are more ethological than using a flash stimulus, but they're clearly not the same as just viewing the real world in any given instance. So we're still not quite all the way there, but we're making our way towards a more ethological stimulus. And I think one thing, if you take one thing away from this talk, I want you to remember that it is bad to just go and use the same stimulus as the first person that did something. So that's actually happened a lot in this field recently. A lot of people have gone, when they start to do these studies, the first thing they do is say, well, I have to use the good, bad, and the ugly because that's what the first study was. But the problem is that means they're only studying the good, bad, and the ugly. They're not moving towards this. By studying many different movies, even if it's ridiculous things like Point Break or something like that, you just add in some random movie that you like, it's still adding new information to the field by increasing the breadth that's being studied. And you may miss or find new things based on what stimulus you're choosing. You don't have to just do this across subjects, though. We can actually use movies as a common currency across different species. So Mantini, who's in Wim van Duffel's lab up in Boston, they ran a study comparing humans to rhesus macaques. So what they did was, the first stage of the study was the same as I've been talking about before. You take individual voxels within the brain and you compare the responses across viewings, and you compare our voxels within the same brain to each other, and you kind of get a map of correlation similar to a resting state correlation. You do that for both the monkeys and the humans. But then what you can do is you can actually take those same ROIs and start comparing ROI 1 to all of the ROIs in the human brain, ROI2 in the monkey brain to all of the ROIs in the human brain. And you can kind of get a cross-species map of correlations. And this can try to look for functional homologies across species. These aren't saying that these are anatomical homologies, but at least they're processing movies in the same way. And what they found was some interesting results. So if you just look at the within-species correlations, you see that the human subjects really do a good job of reproducing each other. The monkeys also are showing a good job of reproducing each other. And then when you compare individual regions across areas, you find some interesting things. So I'm going to show you two examples in a moment, but you do find areas that map very well to, say, an anatomical homologue of each other. So MT goes to MT in the human. But you also find areas that don't map specifically from the macaque up to the human in the way you might expect. So shown here, this is a little bit of a complicated figure, so I'm going to try to walk you through it. They took the right PITD, which is, let's see, right here. It's this area here on the macaque. It's in the temporal lobe. It's posterior. It's dorsal. That's how they named it. And they compared that to every voxel in the human brain. Okay, so now down here, this is a human flat map. These circles represent the anatomical location that you would expect these areas, you'd expect the PITD to fall if it was just a scaling up of the monkey brain. So if you scaled the monkey brain to the human brain, this is where you would expect it to fall, right there. However, what you see is that the activations, the similar responses, actually occur quite a bit outside of there and are diffuse. Similar thing if you try the area more anterior to that. So this area here, this is the anatomical homologue of the CITD, but the area it activated is the ATOS back here in the humans. And so you can do, there's a lot of areas that did match, but you can also see areas that actually moved in a different way, suggesting that the functional homologues may not be the exact scaling of the monkey brain to the human brain. And this can start to probe and ask questions like, oh, okay, I care about this particular region in the human. You may be able to use a study like this to target your monkey studies a little bit better, where you might want to put electrodes. So you don't just scale down the human brain to a macaque, you can actually look for that functional homologue using something like a movie stimulus to target your region. Similar type of thing, we've done a similar thing looking across monkey species. So we can take the responses in the macaque brain, get each voxel, and then we can calculate for each voxel, across the whole brain, we can calculate a independent component space, and we get individual areas, we get these regions that are kind of responding in similar ways to each other. And then we can compare the time course of that similarity to the variables that we coded. We see that this particular group of voxels are all responding similar to each other, and that similarity maps well to contrast. And then we could do a similar thing across marmosets, a small New World monkey that diverged from the Old World monkeys quite a while ago. So we start getting a bigger map of the homologues across species. And we can do this for a number of different ICAs. And what we see is that there are similarities across the species where these two ICs are both responding to contrast. These two ones are responding to motion. We can pick out the face patches in both species. And then what we can do is we can start taking the areas that we found in these ones and we can directly compare them, like the Mantini study, to the individual voxels across species. And then finally, another way that we can get at this is to start comparing across methodologies. So I've talked a lot about macaques in this particular talk because that's one of the things I use. But the macaques allow us to use more invasive studies, right? And so one of the ways that we can actually get at that is to start comparing the responses of neurons, individual neurons in the brain, to whole brain activity using movies as a common currency. So we can take recordings from a particular electrode. In this case, I'm going to talk about one of the phase patches again, because we've been doing that, and compare it to responses in the fMRI. So we take the responses at these two different electrodes, and then we run the normal movies get viewed in both electrophysiology and movies, and then we can take the responses of let me see. So in green is the response of a given electrode, so a neuron. And then in white here is voxel 1. And you can see it's pretty well correlated. And then down here actually is voxel 2. The correlation in white goes way down. And so we can do this across the entire brain. And what we get is a map of correlations for each neuron across the entire brain. So this kind of is a way of multiplexing the response so that we can now kind of expand up to a bigger brain and see what areas, I don't want to say connected, that's not the right word here, but maybe influenced in similar ways. And so what we did was, we did this for a set of 60 neurons that were all recorded in the same phase patch. We compared them to the responses across the entire brain in fMRI, and then we clustered the responses, the correlations that we saw in the brain. And what we saw was 3 different clusters of brain maps within this very small patch of cortex. And what we found was that so cluster 2 is really activated by the faces. These green areas here are the face patches, and you can see these strong red correlations within the face patch system here. But then you have these other ones where there's this strong negative correlation with early visual cortex, or this strong positive correlation with early visual cortex here. So we get these very different maps from these neurons that are all within 100 microns of each other. So these neurons are very close together to each other, but they're responding in very different ways to the point where we can create these different maps. So this starts to suggest that we can start to understand the, I don't want to, again, connectivity is the wrong word here, because we're not showing that this area is particularly connected to those things, but there may be influences coming from early visual cortex through multiple connections that are present for this neuron that are not present in this neuron. And this might start to explain the diversity of responses we see within a given region, particularly within these face patches. So just want to summarize real quick. One of the things, real big take home is that any particular study you're running, whether it's an fMRI or single universe study or optogenetics, the real thing comes down to the trade-offs you're willing to do for being naturalistic versus artificial, and how much control you're willing to give up to go into that realm. The more control you give up, the more natural it is, but then you have to figure out how you're actually going to be able to interpret your data. When you get more controlled, it's easier to interpret your data, but how well it maps onto the outside world becomes questionable. So I showed you that using naturalistic movies, there's ways of pulling out the feature responses. That you can compare different, you can compare the stimulus and the behavior to each other. We can look at temporal information, and we can compare different content across movies. And using this method as kind of a common currency, we can start look at intersubject similarities, interspecies similarities, and between methodologies. Here's some of the main references that I used in this talk, and if you guys have any questions, I'm happy to answer them. No questions? Sweet. Okay. Thanks, guys.